Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today the topic is related to immunology, and today the title of my today's lecture is the phagocytosis and how microorganisms are killed by the phagocytes. In the previous video lecture, I have discussed that how the phagocytosis process begins and how the microorganisms interact with the immune cells. Okay, so if you haven't watched that video first, so first watch that video, because these two videos are linked to each other. So when you are clear about the interaction of the immune cells with the microorganism, so after that then you can understand this video, the part 2. So first watch the part 1, okay, and then after that watch this video, so then you will be clear about the whole process of phagocytosis. So here I will discuss the intracellular digestion, that when the microorganisms are engulfed by the phagocytes, so how these microorganisms are killed, okay. And at the last I will discuss that how then they are released to the environment. Okay, so let's start with the video lecture. This is an overview of the phagocytosis process. As I have mentioned in the previous video, that first these microorganisms interact with each other. Okay, we interact with the immune cells, and when they give a positive signal, so then they are engulfed. Okay, and so how they are engulfed? If I again tell you guys that when there is a positive signal, then so what does they do? Just like this, these microorganisms are engulfed. Okay, a certain portion of the cell membrane of the immune cells, okay, a certain portion of the cell membrane of the immune cells just make a cave like structure, and then these and after that these microorganisms are engulfed, and then a cell membrane is formed again, just like here. So when they are engulfed, so these are known as the phagosome. And as you know that in phagocytes there is a high content of lysosomes. Okay, there are a high content of lysosome. So this phagosome then get attached with the lysosome, or the different lysosomes attaches with the phagosome, and it form a structure that is known as the phagolysosome. Okay, if again I repeat, first of all, there is an interaction between the microorganism and the immune cells. And what is the mechanism of the interaction? What are the different receptors present on the immune cell? So I have discussed in the previous video. After that, they are engulfed in such a way that a cave-like structure is formed, and these are engulfed. Okay, these are engulfed by the immune cells. So this is known as the phagosome. Okay, so if here is a these are the microorganisms. So in a phagosome, they are surrounded by the cell membrane of the immune cell, and so this is known as the phagosome. After that, there is a high content of lysosome in the macrophages, or in the phagocytes. So these lysosomes then get attached with the phagosome. Okay, they get attached to the phagosome, and after that they attaches and they releases their hydrolytic enzymes into the phagosomes. Okay, so when they get attaches to the phagosome, so they are known as the phagolysosome. Okay, they are known as phagolysosome, and these lysosome of the immune cell consist of high hydrolytic enzymes. I will now discuss each and every enzyme in detail, that what enzyme has specific function. Okay, so then these lysosome releases its enzymes to the phagosomes, and after that, because of that enzyme, that microorganisms is killed. Okay, and after that, when it is killed, so then it is released to the environment. Either it is directly released to the environment, or it is presented over the over the immune cells. Okay, so each and every step I will discuss the in detail. So now I will discuss the intracellular digestion, that is different enzymes that are involved in the intracellular digestion. Okay, so the the lysosome consists of these enzymes. Okay, so the first enzyme is the lysozyme, and lysozyme is a special type of enzyme that is involved in the breakage of the peptidoglycan layer. As we know that the gram positive and gram negative bacteria consist of peptidoglycan layer in its cell wall. Okay, so what does this lysozyme do? It hydrolyzes the peptidoglycan layer and it just destroy its structure. So by destroying the peptidoglycan layer, if I just write PG, okay, so it is peptidoglycan layer. So by destroying the peptidoglycan layer, a bacteria can easily be killed, and the different other enzyme can get access to the DNA of that microorganism. So lysozyme, what does it do? It completely destroys the peptidoglycan layer. The second enzyme is the phospholipase. What does phospholipase do? It completely destroys the phospholipids of the microorganism. As we know that in the basically the cell membrane of the microorganism or even the eukaryotic cells are composed of phospholipids. So what does this enzyme do? It hydrolyzes the phospholipids and completely destroy its structure. So by destroying the cell membrane of a microorganism, it can easily be killed because most of the vital functions are performed by the cell membrane of the microorganism. The third one is the ribonuclease. What does this enzyme do? This enzyme destroy the RNA of the microorganism and destroy the RNA of microorganism, the RNA and the ribosomes. Okay, so by hydrolyzing it, so by destroying the ribosomes of the microorganism, it can no longer synthesize protein, and because proteins are very essential for the survival of the microorganism, so when there are no ribosome and there are no RNA, so neither of it will perform function and it will die. Okay. The fourth one is the deoxyribonuclease. As the name indicates, these enzymes hydrolyze the DNA. Okay, it hydrolyze the DNA of the microorganism. Okay, if I write the DNA, so it hydrolyze the DNA. So by destroying the DNA, so obviously the microorganism can no longer survive. And the fifth one and the most important is the proteases. And proteases are the enzyme that destroy the proteins. Okay, they hydrolyzes the protein and convert it into amino acid. Okay, so this protein can destroy each and every protein of the microorganism. So by the action of these enzymes that are present in the lysosomes, the microorganism can easily be killed. Okay, so keep this enzyme and its function in your mind. These are very much simple. Okay, now the sixth step is that how microorganisms are killed. So another function of the lysosome is that another function of the immune cells is that it can form superoxides. Okay, it can convert the oxygen, it can use the oxygen, and these consist of different enzymes that is present in the lysosome, in which the original oxygen that is taken by the immune cells can be converted into superoxides. Okay, it can also be converted into hydrogen peroxide. It can also be converted into hypochloric acid and different other superoxide, singlet oxygen, hydroxy radical, hypochlorous acid. These all are very much dangerous. Okay, if I want to show you that how these superoxide destroy the microorganism, so these superoxide are involved in the oxidation of the microorganism, and as we know, it oxidizes each and every component of the microorganism. And as we know that oxidation is very much harmful for each and everything, even for the immune or even for the eukaryotic cell. If there is oxidation in the immune in the cells, so the cells can be destroyed. Okay, oxidation is the rusting of something, and reduction is its possible is its positive. Okay, so if someone asks you that either oxidation is dangerous or reduction, so you will know, you will tell them that oxidation is very much dangerous. So what does it do? the superoxide have the ability to easily oxidize each and every component of the microorganism. So the enzymes, these enzymes are present in the lysosome of the immune cell. So what does this enzyme do? It uses the oxygen that is taken by the immune cells for respiration. So this oxygen can also conduct respiration, as well as this oxygen can be used to be converted into superoxide or hydrogen peroxide. So these all are very much dangerous, and it can easily oxidize the different essential component of the microorganism. So by oxidation, these component of the microorganism can no longer perform its basic function, and they are destroyed. So by not performing its basic function, these are easily killed. These microorganisms are easily killed. Okay, so keep this fifth step in your mind, that intracellular digestion can also be performed in this type of way. The sixth type is of the intracellular digestion is, if I write sixth type, okay. So this is the sixth type, and it is known as the reactive nitrogen intermediates. The reactive oxygen, it is also known as the reactive oxygen intermediates that I have discussed in the previous video. Okay, those are known as the ROI, reactive oxygen intermediate, and it can also produce reactive nitrogen intermediates in the form of nitric oxide, nitrite, and nitrate, and it can kill the microorganism. How they kill the microorganism, I will now discuss it. The enzymes that are present in the lysosomes have the ability to manufacture nitric acid, nitric oxide, nitrate, and nitrite and nitrate from the proteins of the from different proteins. So what does they do? They uses the amino acids. First they cleave the proteins, and after that first of all they cleave the protein, and amino acids are produced, and after that those amino acids are then further metabolized, and nitric oxide and nitrite and nitrate is produced. Okay, and these are known as reactive nitrogen intermediate. So what does these intermediates do? They interact with this part of the electron transport chain. There is a high content of iron that is playing an essential role as a cofactor in the electron transport chain. These all are the carriers of the electron transport chain. Okay, protein electron carriers. So, and in these carriers, iron is an essential part. Okay, if there is no iron in this electron carriers, so these carriers can no longer perform its function, and they can no longer respirate. Okay, so what does these nitric oxide and nitrite and nitrate do? These intermediates combine with these part of the electron carriers. Simple, they combine with the iron portion of the electron carriers. So by destroying the iron component, or by binding it with the iron component of the electron carriers, so this iron can no longer accept the electrons. Okay, so by not accepting the electron cannot move in the electron transport chain, and there will be no respiration. Okay, and respiration is very much important for the microorganism, because they are producing energy by the process of respiration. So these intermediates interact with the iron part of these electron carriers and block its function. So by blocking its function, it can no longer respirate, and the microorganisms are easily killed. Okay, the microorganisms are easily killed. So this was all about the intracellular digestion. These were the special type of enzymes that were performing its function, and I think now you are clear about it. And this is the fifth part, that reactive oxygen intermediates are also produced, and they oxidizes the internal component of the microorganism, and by oxidizing it, it can no longer perform its function. The sixth was the reactive nitrogen intermediates. They interact with the electron carriers present in the electron transport chain and block its function, and the microorganism can no longer respirate, and they are killed. Okay, so this was all about the intracellular digestion, that how microorganisms are killed. The last part of this video is that exocytosis, how microorganisms when they are killed, so how they are released by into the environment. So first of all, keep this thing in mind. In case of neutrophils, okay, just in case of neutrophils, what does neutrophils do? They are not as professional phagocytes as macrophages and dendritic cell. So what does these neutrophils do? They simply destroy the microorganism and they convert it into small pieces, and they directly release it into the environment, into the bloodstream. Okay, these microorganisms are the debris of the microorganisms are directly released to the bloodstream or to the internal spaces or to the lymph nodes. Okay, so they are not such responsible immune cell, but they just destroy it and they releases the debris into the environment. This is just in case of the neutrophils. Okay, or and in the case of the macrophages and the B cells and the dendritic cell, what does they do? They first they destroy the microorganism, and after that, by the debris of the microorganism are then travel to the endoplasmic reticulum, and after that, if I write here, these are the debris, and after that these debris are transported into the endoplasmic reticulum of the macrophages, the B cells, and the dendritic cell. After that, there are special type of proteins that are known as MHC2 proteins, MHC1 and MHC2 proteins that are present, that are performing a specific function, and now I will tell you. So what does they do? They are present in the endoplasmic reticulum. So these debris then binds to the MHC molecules. Okay, MHC molecules. Okay, sorry, MHC molecules, either MHC1 or MHC2, but mostly MHC2. Okay, and these are special type of proteins that interact and that bind with the debris of the microorganism, and after that, in a unique manner, they get out of the endoplasmic reticulum and they displace these debris over its surface. Okay, now this is the proper function. This is performed by the neutrophil. They are just released to the environment, and this process, these debris travel to the endoplasmic reticulum, and after that they binds to the proteins that were present in the endoplasmic reticulum, and these proteins are known as MHC proteins. Okay, and after that these travel to the plasma membrane. Okay, and they are these antigens and these debris are presented over the surface of the surface of the immune cells, and this is only performed by the B cells, the macrophages, and the dendritic cell. So the plus point of this process is that these debris are not introduced into the bloodstream. They are just processed in the unique manner, and these are presented into the other immune cell. So then after that, the immune cells of the adaptive immunity, the T cells, okay, mostly the T cells, then the T cells come here and they binds to this debris and then further recognize that which type of antigen it is. Okay, so T cells, when it is released to the environment, so T cells can hardly recognize these antigen and bind to it. But when it is present on the MHC and it is presented in a specific manner, so then T cells can combine and it can recognize this microorganism in a unique way. Okay, and then after that, how they interact and how they convey this message. So it is a topic for a new lecture. Okay, I will discuss it, and then how T cells are activated. Okay, so keep these points in your mind, that and this was all about the phagocytosis process. In the first, I discussed that how microorganisms are interacted with the immune cells. After that, when they are engulfed, so how they are presented, how they are digested by different processes, by different enzymes, and after that, how they are introduced and how they are exocytosed. Okay, so this was all about the phagocytosis process. and I think you have get it. If you have understand this video, so like this video, share it with your friend, and subscribe to my channel for more interesting videos related to science. And if you have any questions, so just ask me in the comment box, I will answer you there. Okay, thank you very much, God bless you.